Welcome everyone to our Lunch, Learn, Link seminar sponsored by the Sidney Kimmel Comprehensive Cancer Center and the Maryland Cigarette Restitution Fund. Today we have a real treat. John Carethers from the University of Michigan is with us to talk about colon cancer and the breadth of his research and maybe some stories about how he got there. So it will deal with disparities, but also inflammation and the genetics of colon cancer. So I'm really happy that all of you could attend. Welcome to our online viewers as well from University of Maryland and Howard University. And for those of you in the audience today, Nicole will be at the side with tickets for your pizza. So if you have a ticket, you can grab a piece of pizza at the end over in the gallery. We'll remind you. It's wonderful pizza. So today, John Carethers, he's the C. Richard Boland Distinguished University Professor and John Searle Professor of Internal Medicine at University of Michigan. He's going to talk about cancer. He is a gastroenterologist, so a very practical view, maybe population-based view of the issues. And he just has a wealth of literature that he's participated in. So I think you'll find this a treat. So I turn it over to John.
Thank you, Norma, and thank you for having me here at Johns Hopkins. It's certainly a pleasure, and I thank you for a wonderful dinner last night. I'm kind of an accident in getting into the disparities work. When I started out for my training, I was more singularly focused on just trying to understand colon cancer. And by the way, my first project in my mentor's lab was on programmed cell death. And then that was my major project. And I even wrote a grant on it, but we never published anything on that. Then I had this minor project that we didn't even know what it was yet. It turns out it was DNA mismatch repair. I just happened to be in the right place at the right time back when I joined my mentor's lab in 1992, 1993, before the first papers on human colon cancer microsatellite instability were published. And we were, Juichi Sato, who was a postdoc in the lab, would show these gels to my mentor, and these alleles would move around. And Rick would say, "You're doing it wrong. You're doing it wrong." But it was actually microsatellite instability. We just didn't recognize what it was. And then that became my major project that I've been working on for the last 25, 30 years. So it's just weird how things happen. So that's how I started out. And then later, it wasn't until someone asked me to write a review on cancer disparities, and this was probably late 90s. And I'm like, why would I write that? I mean, I think they assumed I worked on cancer disparities, but I didn't at that time. And then when I started looking through the literature, there wasn't much data out there. So we've learned a lot since then. And I'll hopefully show you some of the things we've learned, not only what we've done, which will be more on the second half, but some of the stuff that other people have done epidemiologically to try to understand this better. So I have no disclosures other than NIH and University of Michigan internal funding. So I'm going to talk just brief on, you guys probably know this better than I do perhaps, but the epidemiology of race and colorectal cancer with a handful of slides. And then tell you what little we know about race and genetics in colorectal cancer. There's not much out there. And then spend probably the bulk of my presentation on inflammation and how it affects DNA mismatch repair and how that influences things that we found between race, largely between blacks and whites. And then kind of summarize that and actually give some future directions. So being a gastroenterologist, I focus on the GI tract. And looking at SEER data, this is a paper we published a couple years ago on 7 of the GI tract cancers, and what is the incident and mortality ratio. So this is a kind of an artificial way of looking at is a cancer more common in blacks versus whites when you do these incident and mortality ratios. And there's 2 things that come out of this. One is the racial difference between those 2 races? And second, is there any additional disparity with the mortality piece of this? And so if you, I'm going to use the mouse here. So esophageal cancer, if you look at the black to the non-Hispanic white ratio, it's 0.75, meaning whites have more esophageal cancer than blacks, okay? And that ratio is pretty much between incidence and mortality about the same. So probably it's from the beginning, not during later when you get treatment and other things. And this is largely because of the increase in adenocarcinoma, particularly among whites with gastroesophageal reflux disease. If you look at stomach cancer, for instance, you see that incident ratio between blacks and non-Hispanic whites is 1.77, meaning blacks have more stomach cancer. And then there's an additional bump up between the mortality ratio, meaning the things that happened, there's probably other things that are influencing the worst mortality after the incidence is taken into consideration. Pancreatic cancer, similarly, but there's no bump up on the mortality ratio. Small intestinal cancers, which are fairly rare, and that's a whole nother discussion we can have. Why do you have 18 feet of small intestinal bowel and you rarely get cancers, and you got 4 or 5 feet of colon and you get tons of cancers? That's a whole nother story. But the incident ratio and mortality ratio are about the same, higher in blacks versus non-Hispanic whites. Anal cancers, they're pretty much uniform. It's largely HPV driven. Colorectal cancer there's a disparity in the incidence as well as a bump up in the mortality based on these types of ratios, et cetera. So you can get a feel for what's going on in the GI tract and how there's some disparities, at least between blacks and whites. If you look at cancers and how we screen, because screening is still the ultimate prevention modality, this is the latest data from the American Cancer Society showing that colorectal cancer is going to affect about 145,000 people this year. So it's not the most prevalent cancer. It's the 3rd most prevalent cancer. But it's the 2nd deadliest behind lung cancer, estimated 51,000 deaths this year. Now, it's very interesting. For breast cancer, we have mammograms and self-exams. Cervical cancer is pap smear. Prostate cancer, rectal exams. PSA is the controversial thing. Colon cancer, we've invented 7 or 8 tests because it's so deadly. But now we classify these into tiers. So our first-tier tests are usually the fecal immunohistochemical test and colonoscopy in the United States. But in Great Britain, it's FlexSig, and in some other countries, it's just the FITS. And we have 2nd and 3rd tier tests that we do that have different levels of efficacy. The FOBT is now relatively obsolete because FIT has just kind of replaced that. And barium enemas are, most radiology people did not like to perform those anyway, but their sensitivity is actually pretty poor. So this is a 2nd deadliest cancer, lots of tests to go after, although that is slowly getting organized in the GI literature as well. There are a number of deaths, as I mentioned, that are different between, at least with the accurate data between blacks and whites. And without going through each one of these cancers like I did on the first slide, I just want to point out of all these GI cancers, colorectal cancer is slightly over 40% of all the GI tract cancer burden, 40%. And if you look at the overall mortality to incidence ratio for colon cancer, it's about 37.1%. Thank you. But if you look at the black mortality, it's 40.7%. Meaning that it's higher by those ratios. So data from the American Cancer Society shows these rates per 100,000 of the population standardized to the 2000 census. So you look at overall, these rates for male and females are in the order of 35 to 45 per 100,000, with the deaths in the order of about 12 to 17 per 100,000. But that varies by race. In every category, blacks were higher than whites. Certainly much higher, whites are even higher than Asians. The data has been slowly been gained for American Indians and Alaskan Natives. And I think and this is just my own theory, I think if you looked at data 10 or 15 years ago, those numbers were much lower. But I think they're living longer and not dying earlier. And now you're seeing the manifestation of the cancers coming out there. A colleague from Howard, I don't know if he's online or not, published a paper looking at young onset cancers. This is another hot topic in the colon cancer field. And even looking at people aged 20 to 44, and this is the data that's published in 2016, so that's the latest data we have, shows that even young black patients had a higher rate per 100,000 compared to whites and Asians for the data he had at that time. So some of these things may start earlier. So why is that? Well, we don't fully know. If you look at the trends, for both males and females, the good news is colorectal cancer rates and deaths are coming down. But for males, there's still that disparity ratio. They're both coming down, but they've actually widened. So if you look at the black line, it's starting to level out for that mortality ratio. For females, that line is starting to trend down, meaning they're slowly closing the gap. And although I'm not going to spend a lot of time on this, there's great evidence for intensified screening through navigation can mitigate some of these differences over time, particularly at the level of the screening. If you look at stage of survival, we're still seeing differences. So this is the data, again, just published a couple months ago from data from the American Cancer Society. There are slightly less localized and regional for blacks compared to whites for colorectal cancer, meaning those are the ones that typically are cured, but higher distance, which are less likely to be cured. And if you look at the survival rate for every single stage, it was slightly less. And this ranged from a few percent to as much as 7 or 8% for that. Now this next slide is a figure that we put in a review that I wrote summarizing data from screening populations and then looking at what happens between, in this case, blacks and whites, or the blacks, that's just blacks in the general population. And basically, if you look at the proportion of colorectal cancers that happen below the age of 50 when we start typical screening, and these are the screening recommendations up until recently, that you would have 5.5% of all colorectal cancers under the age of 50, but essentially 94.5% cancers above the age of 50. And most people would think, for the most part, that those would be more hereditary-like cancers or the unfortunate young person who developed those. And we're seeing an increase. Now, that number is approaching 10% over time. If you look at African Americans using the same data, with the exact same data, the proportion under 50 for that target population was double. It was 10.6%. But if you moved the screening of African Americans to 45, you bring it down to 5%, right in line with the original recommendations. And this has fueled probably debates over the last 10 or 15 years by calling out that population for screening slightly younger. And several organizations have proposed that, and up until 2017, none of that had actually gone into effect. There were a lot of proposals by a number of different organizations, some pushing as young as 40. But in 2017, the U.S. Multi-Society Task Force on Colorectal Cancer Screening, which is a consortium between gastroenterologists, radiologists, cancer specialists, et cetera, this was the first time ever they recommended a specific target population other than high risk familial disease to move it to 45. Now, has the U.S. Preventive Services Task Force enacted that yet? No. Can you get it paid for? Some limited things, but usually it's the U.S. Preventive Services Task Force that usually goes into CMS regulations, and so that's when you usually get it paid for. But there's a ways around that. Whether those are fully legal or illegal is another story. But that's what the data shows. The other thing is there's a lot of data just summarized here that African Americans present anywhere, depending on the papers you read, 0 to 8 years younger compared to Caucasians. And what that implies, and again, showing that there's a slight uptick in colon cancer under the age of 50 from Hassan Ashkarab's data, that if you start screening at age 50 and they've already had a dwell time of a polyp for more years than typical, that shortens the window for screening intervention. So let's say an average person starts developing a polyp at 35, and it takes 20 years for that polyp to potentially do something, and you find it at age 50 and you remove a large polyp. But if it started at 25, you might get the cancer before your typical screening age. So that dwell time makes a huge difference, as does the starting time for when you start screening. I hope that made sense. Now, the other thing is, these are colon cancers. We know that polyps, for the most part, are the precursor to colon cancers. And so there's been some very wonderful studies looking at the distribution of polyps based on race. And I'll show you David Lieberman's work from the CORI database. The CORI database was an NIH-funded database that looks at endoscopy practices across the country. This was one of his earlier papers, but the numbers get bigger over time. This was looking at 67 adult practices back in the mid-2000s, and you can see the numbers of whites and blacks who were in there for a screening population. The bottom line is, if you looked at the polyps that were more likely to potentially become cancer, these are the high-risk polyps. We call them adenomas of advanced pathology. These are ones that are usually greater than 9 millimeters, essentially a centimeter in size, may have a villus component or a component of cancer already in them. That blacks had a 1.5% higher rate of those larger polyps than whites in his study. And very interestingly, if you look at this data over here at the odds ratio, certainly as we age, that ratio went up, which is what we would expect. They used the community practice as one. Academic was slightly less. Maybe people who go to academic practices are less likely to make polyps, who knows. And in the VA population, they were 10% more likely to make polyps. And this is something, having worked in the VA system for 15 years, it seems like vets have a lot of polyps for some reason. But the real data was comparing blacks versus white, women versus men. And black women had a odds ratio of 1.62, black men at 1.16 over their white counterparts for developing polyps, high-risk polyps. Again, kind of confirming the data that we see with colorectal cancer, since these are the precursors. The other interesting thing is that this was published about 3 or 4 years ago from the same group, who have now grown to 84 adult practices and looked at now 325,000 average risk adults. So these were screening. And what they looked at is they looked at the polyps based on race and every 5 years of age. And I have red bars for when there were more polyps in the black population than the white population. So if you look in females, that's true for every one of these 5-year age intervals, except for 70 to 74 for some reason. And if you looked in men, that was true for particularly black men for every age everyone over the age 50. And the reason why he did that was because we knew age was a powerful driver, but he wanted to compare a little bit more narrow to see if that was still true even at those 5-year intervals. And that was true for these high-risk polyps. So the other interesting thing is where these polyps are. And we do know a lot of data that, particularly amongst Black patients, there's a higher percentage of right-sided colon cancer. And so does the adenoma data match that as well? And this was looking at, again, these high-risk polyps, proximal versus distal, and looking at the ratios. So we can skip the age here, which but if you look at whites, there was 50.6% proximal, 49.4% distal, almost half and half based on his study. If you look at Blacks, that there was a 6% differential between the proximal and distal location. Now, why is that important? When we do screening colonoscopy, and there's a lot of data on this, we're very good at clearing out the left colon. The right colon's a little bit worse. And if you look at recognition and survivability from colorectal cancer, it's worse on the right side versus left, despite screening. Even though colonoscopy is the best test, that is the gold standard. But there's data showing if you did tandem colonoscopies, whoever volunteered for that, that there's still a 5 to 6% miss rate with colonoscopies, even though that is the gold standard. And there's several papers now describe that the risk of subsequent colorectal cancer formation and mortality is worse on the right side, even with screening compared to left. So that if someone's developing polyps on the right side all the time, in which they tend to be flatter, a little more flesh colored, they're harder to see, maybe the quality of the prep is a factor, who knows, but the outcomes on the right side are actually different than the left side of the colon. The other thing is these interval cancers. So this gets at someone who's been screened, and then between the normal screening interval appropriate for what was found or not found at the beginning has another screening. But in between those two time points, people develop cancers. These are still somewhat rare, and there are some biological differences. They tend to be more microcyclically unstable, but some of these are could be the quality of the endoscopist, as well as the ability to observe the lesion, perhaps a precursor lesion that was very difficult to see. So in this particular study, which just came out in the Annals a couple years ago, it turns out almost 53% of Blacks who had screening had physicians who had lower polyp detection rates than versus 46% of Caucasians. So that means the quality of the physician may have made a difference. Now, they did standardize this later on. So if you look at the interval risk probability, I can tell you this solid black line is whites, and this top line is Blacks. And this is the cumulative probability of an interval colon cancer. So over time, and this is in months, that if you were Black, you were more likely to have an interval cancer. Now, the scale of this is pretty small, but still it was 5.8% versus 7.1% with a hazard ratio of 1.31. Interestingly, the authors recognized that the quality of some of the endoscopies was a little different, and we do standardize this as a place. So people who have low polyp detection rates have an intervention to raise their polyp intervention rate. So we're trying to do this right at least our institution. I hope that's done at every institution. But adjusting for the higher polyp detection rate did not remove the disparity. Interesting, at least in this study that was published in the Annals. So that's my general epidemiology. Now, let me tell you about race genetics. I only have a couple slides on this, and that's because there's not much known. So first of all, what about hereditary genetics? We know a lot about the high-risk syndromes. These include things like FAP, MYH-associated polyposis, the polymerase epsilon delta polyposis, Lynch syndrome, which is the most common one. That affects 3 to 4% of the population who get colon cancer. There's some other rare runs. And as best we know, there are founder mutations for some of these syndromes, but they're seen in essentially every race. We see them in Saudi and Middle East families. We see them in Finland. We see them in the United States. We see them in South America. So FAP is described on every continent. Lynch syndrome is found on every continent. There's some exceptions with founder mutations, but it's all over the place. And it does not appear to be focused in at least one racial or ethnic group, as best we can tell based on the data that's out there. Now, what about somatic genetics? So this is a slide from the TGCA data set that summarizes about 30 years of genetic work in colon cancer. So let me just walk through this quickly because it helps explain the next slide. This upper graph on the y-axis shows the mutation rate. This is inside the cell's genomic DNA. So this will be a cancer cell DNA. And the x-axis has the tumor site, the microsatellite instability status. I'll walk through that in a little bit. The CIMP status, this is CpG methylated phenotype status, and MLH1 silencing. And if you notice, almost all the red bars overlap each other because they all cause the same thing. But just to suffice to say, that is about 15% of all these tumors. And these are all hypermutable tumors, meaning that they have anywhere from more than dozens to 1,000 mutations. This is a logarithmic scale. So that's 15%. And essentially all of them are the ones that have a microsatellite instability phenotype or genotype. The other 85% follow the classic Burt-Vogelstein model, in which they are non-hypermutated and they follow a series of genes that drive its pathogenesis. So if you look at the non-hypermuted, the classic APC, P53, KRAS, PI3 kinase pattern, you'll see here, there's a handful of genes that are very common in almost all somatic mutations for these tumors. And then there's a long tail that might individually be present. In the hypermutated MSI positive tumors, you notice that the tail looks different because most of these genes, for the most part, have coding microsatellites in their genes, and they get frame shifted. So they take out ACVR2, which is the activin type 2 receptor. APC has a couple of coding microsatellites in it itself. The TGF-beta R2, another one of the TGF-beta superfamily genes. BRAF doesn't have a coding microsatellite, but it segregates with this group. That's one of the few that doesn't. And even mismatch repair genes, MSH3 and 6, have coding microsatellites in them. And you notice that the percentage is more uniform across because it's largely based on the structure of those genes that get affected compared to the non-hypermutated group. Now, I went through that. I should say one other thing. If you look at this hypermutated group here, this is in the same order of the tumors listed here as in this inset box. And you can see, for the most part, these gray boxes, it's basically methylation of the mismatch repair gene MLH1. There's a few ultra hypermutated driven by the polymerase epsilon mutation there, and that doesn't really overlap with the MLH1 hypermethylation. So I mention that because this slide has all the data we have on the least difference between African Americans or blacks versus whites. So a colleague of mine, Sonia Kupfer's group at the University of Chicago, published in 2015, largely looking at Lynch syndrome families. So these were African American Lynch syndrome families. And what they found was there was a subtle difference between the type of mutations you found in these mismatch repair genes, that 2/3 were specifically MLH1. Typically, MLH1 is more about half, 45, 50%, not 66% of the mutations are found in Lynch syndrome. So they found 2/3 were MLH1, and they found some novel mutations that may have come from various ancestral backgrounds. But the cumulative risk for cancer was exactly the same as those from European descent. And so she just closed that article by saying there's probably some genetic diversity in the mutation spectrum, but the phenotypic outcomes were pretty much the same. So that was it. And that's about all we know, the differences. Now, what about somatic mutations? And there's been a lot of theoretical areas on this, but not too much to back things up. There was one paper published in PNS by Sandy Markowitz's group at Case Western, which found through whole genome sequencing of multiple tumors that there were 2 or 3 genes that were uniquely mutated in colorectal cancers from Black patients. And they focus on EPHA6 and folliculin. And those, if you remember, germline folliculin causes Birt-Hogg-Debay syndrome. It has nothing to do with colon cancer. That's a kidney cancer and a lung disease. But they found these as potential unique driver mutations. I have not seen any follow-up of this and any verification of this, although I heard one from being on an external advisory board that someone did some sequence of a handful of tumors and they found some of these same genes. But I haven't seen any other publications on this to date. These were deleterious mutations. If you look down in this box here, they were missense, and one was a splice for the EPHA6, and a couple of these were insertions or nonsense mutations that were presumed to be driver somatic mutations. So I'll say two more things about this. One is I think this needs to be verified, and we haven't seen any verification to date. And second, this group, nor anyone else, has proven that these are true driver mutations for the tumor yet. So this is out there, but there's still a lot to know. And that's it. So let me spend the bulk of my time on inflammation and repair. Now, we know that you can take someone who's presumably genetically susceptible and give them an environmental exposure, and the outcome for some of those groups will be a cancer, depending on what that environmental exposure is. It's a gene-environment interaction, whether it's a pro-carcinogenic virus or a change in your microbiome because you are what you eat, you're feeding the bacteria down there that may make certain products and digest certain products. Now, these things can cause various levels of inflammation. And I'll argue, even if you look at the entire GI tract, many of the cancers in GI tract are a result of inflammation. GERD and adenocarcinoma. H. pylori induces inflammation in the stomach, gives you stomach cancer or maltomas. Inflammatory bowel disease is a high risk for developing small intestinal as well as colon cancers, both Crohn's and ulcerative colitis. And certainly, I've had people who had even non-HPV anal cancers from because they're incontinent and repeated inflammation and damage develop a cancer, sometimes a skin cancer or early adenocarcinoma. And certainly, the liver and pancreas, repeated bouts of inflammation with pancreatitis puts you at risk, and same thing with the liver. And so inflammation can be a big driver, at least along the GI tract, for many of these cancers. If you look at what are the risks for colorectal cancer, I always classify these into modifiable and non-modifiable. Non-modifiable, you can't change your age unless you go back in time somehow, and let me know if you can do that. You can't change your family history and your race. I mean, those are really the inherited factors. But things that can modify, and we know the environmental exposure include diet and diet supplements. That's fueled by socioeconomic factors and healthcare access, the use of tobacco, the use or non-use of NSAIDs, those types of things. And I would argue that all those things, to some degree, directly or indirectly, will affect the level of inflammation that you have. Okay? Now, this is a study from Steve O'Keefe, published a few years ago, which is kind of a mind-blowing experiment. What he did, and this was to assess what happens in the colon by your diet. And what he did, he took African Americans, blacks, from a cohort here, and then he took some rural Africans from Africa. And now, keep in mind, the epidemiology of colorectal cancer in rural Africa is pretty low. Okay, it's below our radar. Of course, the rates I just showed you are very much higher in the United States. And what he did is a diet swap. He took the rural African diet, gave it to the African Americans here, and then he gave this westernized diet by changing the lipid content and all this other stuff, and gave it to the rural Africans. So the African Americans had a much higher fiber, low fat, whereas the rural Africans now ate the nice high fat, low fiber, westernized diet. And then what he did, he gave him this diet, had a wash-in period, and then he took pre-biopsies before the diet change and biopsies after the diet change, and to see what happened in the colon itself. This is just fascinating. So on this left graph, the series of graph here, Ki67, which stains proliferating cells. So if you look at the African Americans on the original biopsy, this is the level of proliferation they had. And when they went on the rural African diet, the level of proliferation was cut in half. And on the Africans who went on the westernized diet, their level of proliferation doubled. This is just from staining the cells in the crypts of the colon. If you look at intraepithelial lymphocytes, the level of inflammation, it dropped not quite in half for the African Americans on the rural African diet, but statistically went up the level of inflammation in the colon on this westernized diet for these rural Africans who weren't used to this. And if you look at macrophage markers, similar thing happened. Now, if you look at what happens to the metabolites, because you're probably changing the microbiome in this, butyrate is a short-chain fatty acid that fuels colonocytes. It's one of the few food sources for colonocytes to be healthy. And we know this because if you take someone and divert their colon from the fecal stream for a while, they'll get a bad pouchitis because there's no short-chain fatty acids in that fecal stream. And the fix is to rehook them up or give them fatty acid enemas. Okay, so it's a fuel thing. So it's a good thing. But when you switch the diet within 2 weeks, and this is, I think this is diet week 2. I can't remember the label here, but the bottom line is the African Americans, the U.S. people, had much higher level of butyrates after that 2-week period, and the rural Africans who were eating the westernized diet lowered their butyrate levels. Okay, so the good short-chain fatty acid, the ratios change. Now, interestingly, we make secondary bile acids as well, and it's been proposed that secondary bile acids, which require bacteria input from the primary bile acids that we put in our enterohepatic circulation, which are modified by the bacteria to secondary bile acids, is a potential pro-carcinogenic compound. No one's 100% proved that, but there's been some associations. And so they looked at the secondary bile acid, deoxycholic acid, and the opposite thing happened. African Americans, the level of deoxycholic acid went down on the rural African diet, and for the rural Africans on the westernized diet, it went the opposite direction. So the bottom line is, on this new diet, the African Americans increased the butyrate, decreased the deoxycholate, and just the opposite happened. So this was within 2 weeks. Can you imagine being on this diet for 30 or 40 years, where you're altering the proliferation? So if this holds up for a long period of time, so I say this because, some of the things I'm going to tell you about is we find this in a certain race, but race may just be the proxy for what we eat and what we make. Okay. The other thing I'll point out in this is that there's been a number of really good studies, principally by this guy Gallon in Europe, showing that the level of inflammation can predict your outcome better than classical staging. And this was a paper he published in 2006. He's published multiple papers since then, showing that, typically stage 1, early stage, great survival. Stage 2, a little worse survival, but not too bad. Stage 3, you drop off. That's why you need chemotherapy for that. Stage 4, survival is like 5%. It's bad no matter what, because it's metastatic disease. But if you break it down by the level of T cell infiltration, and this is CD3 stains T helper and all T cells, essentially. And he counted the cells in the center of the tumor, that's CT, and the invasive margin, that's IM. And if they were high, no matter what stage you were, you had pretty good survival. Stage 1, 2, or 3, all bunched up here. If you had low CD3 counts at the center of the tumor or invasive margin, you had lower survival, irrespective of the stage. And of course, stage 4 just did poor no matter what you did. But it did point out that the immune system is very important in this. Now, let me tell you a little bit about DNA mismatch repair. Many of you probably know this, but I mentioned Lynch syndrome earlier. DNA mismatch repair is an enzyme system that's evolutionarily conserved. This system is present in single-cell organisms because it repairs DNA. It's a little bit more complex when you get to yeast in humans, but it's essentially the same system. If you go back to yeast and bacteria, they're called the S and L proteins. And then later in humans, we call it the mutated S homolog, MSH, or mutated L homolog of these proteins. So these are the proteins that are in almost all living organisms to some degree or another. Of course, the level of the protein is a little more sophisticated in some organisms. And they work together to repair DNA. Principally, the main function of these proteins in human DNA is to repair post-synthetic mistakes. And these are the level of the single nucleotide as well as at these repetitive sequences we call microsatellites in the DNA. Okay, it's called mismatch repair because it fixes mismatch, but it's called microsatellite instability because it also fixes microsatellite DNA. So it requires several proteins to work. Complete loss, if you take out MSH2 and MLH1, which are the two most common genes in the germline of Lynch syndrome patients, that's why you get early onset cancers. Okay, PMS2 will do it, but it's slightly later onset. The partial loss is because there's for MSH6 and MSH3 is because their biochemical overlaps slightly. These proteins interact as heterodimers. So MSH2 will pair with MSH6 or 3. And in humans, MLH1 pairs with PMS2. In the yeast, it's called PMS1. But it's called PMS2 in humans, so that's all you need to know. They work at heterodimers, and when one is not there, the other one becomes unstable unless it has a viable partner. So MSH2, since it can pair with 6 or 3, will be around even if 6 is missing because it can pair with 3. But if MLH1 is missing, PMS2 will also become degraded. So this is an example of this. This is an immunohistochemistry over here showing MLH1 is missing, but you see PMS2 is also missing because its binding partner is gone and by itself becomes unstable. In this case, MSH2 is present, and you see MSH6 is present. If MSH6 was missing alone and MSH2 was there, this is clearly an MSH6 mutation. Okay? So the loss of one partner causes instability of the other partner. Of course, when we look at these at microsatellites, which are repetitive sequences 100,000 times throughout the genome, this is what I was looking at when I was in my mentor's facility that we didn't understand when these alleles would contract or expand in length. That's called microsatellite instability. And this study, which Paul Motrich did in bacteria way back in the 80s, he received the Nobel Prize in 2015 for understanding this process. This is how the system works. So I said 2 compare with 6 or 2 compare with 3. Rick Fisher showed that this acts as a clamp. It actually surrounds the double strand of DNA and actually can detect defects in the DNA that interfere with normal Watson-Crick hydrogen bonding base pairs. So here's drawn a CT mismatch. It's a single mismatch. So 2 and 6 would recognize that. They hydrolyze ATP to close, kind of like almost like an actin-myosin muscle fiber. And then it caused a second group, MLH1 and PMS2, to signal for repair, or if the repair is futile, signal cell death. Now, it can also do it at these microsatellite sequences. What I have depicted here is a tetranucleotide sequence. This is a repetitive sequence of AAG, AAG, AAG. And you can see any AAG can pair with its partner TTTC on the other strand. And if they don't perfectly pair, I call it the broken zipper model, where you can loop out one because any of those AAGs can pair with any of those TTTCs. So if it doesn't line up exactly, you got to loop out of, in this case, 4 nucleotides. If it was mononucleotide, it'll be 1 nucleotide sticking out. This can happen on the template strand or the newly synthesized strand. And so that will tell you if you get a contraction or expansion or this insertion deletion loop. And so the system will repair both of these types of lesions. So what are some common defects with some common features of defective mismatch repair? In the sporadic cancers, it's seen in 15% of all colorectal cancers. So pretty common. The tumors display microsatellite instability. If I was able to take DNA from those tumors, I can show you that there's microsatellite instability. There's the shifting of these, the length of these microsatellites. And as I showed you in this previous slide, you can lose DNA protein expression. And in most cases, in sporadic, it's due to hypermethylation of the MLH1, which will take it out from being transcribed and translated, and then you lose the expression of MLH1. 70% of these tumors tend in the proximal colon. These are right-sided tumors. In fact, when I started studying this disparity, I'm like, right-sided tumors, there's more right-sided in African Americans. It must be higher. Wrong. I'll show you the data on that. They often show a poor differentiation, and 40% of them will make mucin for some reason. Okay, that's over the 5 or 10% that you see in non-MSI tumors. They are hypermutated, as I showed you before, containing 100 to 1,000 mutations because they're not repairing the DNA. So there's a lot more mistakes accumulating in that cancer cell genome. Despite that, they're mostly diploid, and very few have p53 mutations, unlike the classic Vogelgram model. Now, here's the unique thing about the inflammation for these types of tumors. These colorectal cancers that have microsatellite instability attract T cells to it. The loss of mismatch repair causes attraction of T cells. How does that happen? You have these 400 to 500 genes in the genome that have coding microsatellites. They get frame shifted. They make neopeptides that are novel to our immune system. And the cells come to those novel peptides and tries to understand. They actually contain the information. This is why patients with microsatellite unstable cancers do better than those who don't have it. They are also resistant to 5-FU. I can give you a whole nother hour talk on that, but I'm not going to go into that. And they show longer survival, as I alluded to. This is the pathway that MSI tumors go. You start normal. You still have to take out the Wnt signaling, which includes APC, but typically you lose mismatch repair relatively early. That can happen in a germline for Lynch. There's something called Lynch-like, which is a biallelic somatic mutation. But for most cancers, it's sporadic and it's methylation of MLH1. You can get BRAF mutations, not in the germline, but in the sporadic version. You get this chaotic multiple mutation, hypermutatable phenotype, which includes these immunogenic neopeptides from many of these genes, because now they're not forming their full, they're forming these truncated, that essentially cause an immune reaction, and the patient is essentially self-vaccinating themselves against their tumor. Those frame shifts, you can detect the microsatellite instability high. As those frame shifts accumulate, they also acquire expression of PD-1. And this is the trial that Luis Diaz, who was here, showed beautifully that these tumors are susceptible to anti-PD-1 therapy. So that's an induction of protective T cells. This is what these tumors look like histologically. They tend to be poorly differentiated, so not organized like you see in these well-differentiated ones. They tend to have these mucin. This is what this whitish, grayish stuff looks like. And because they're making all these neopeptides and causing the immune system to react, several of these tumors, and a good pathologist can pick this out, will have this, what I almost call Crohn's-like reaction. So Crohn's disease, 10 to 15% will have granulomas. These are not granulomas, but they look like granulomas if you look at them from distance, but they're actually immune T cells nest in the subepithelium. This is the deeper layers. Here's the epithelium, the cancer, and you see these collections of these immune cells that are responding to those neopeptides that are being made from all this frame shifting. It's pretty amazing. This is Luis Diaz's work. It was published a few years ago, showing that mismatch repair deficient tumors had a biochemical response, that's what's in the blue, as well as a radiologic response in terms of shrinking the size of the tumors. performance-free survival was much greater if you had a deficient mismatch repair tumor. So how does this work? So you get alteration of mismatch repair, you get all these target gene mutations, then frame shift induces protective T cell inflammation, and the biological consequences is that you have a lot more prison guards around the prison, and the prisoners can't get out. And their survivability stage per stage is much better. And this is why I think 5FU is bad, because those cells are resistant to 5FU, but the immune cells are sensitive to 5FU, and you're killing off the prison guards, and then you're making the things worse. Can defective mismatch repair explain any of the colorectal cancer differences with race? This is a study we published a few years ago looking at 500, a population-based, this is the only population-based study, so I figured I was at School of Public Health. So this is a population-based group from a collaborator at North Carolina, Bob Sandler, who's collected colon cancer information from 33 counties in North Carolina, and he particularly targeted those that were higher African-American populations. And so he has all this data on diet and all this other stuff that he does a lot of work on. And I just asked him, can I get the tumors and study this? And so we were totally blinded until 2 days before the abstracts were due. It turns out 45% of these were Black and 55% white. And the total MSI prevalence was 11%. And this, in the literature, this ranges, we say average 15%, but it ranges really from about 11 to about 17%, but it averages about 15%. The interesting thing in this group is that 14% were whites, but only 7% were in the Blacks. And we were blinded to this. And they're like, wow, because if you think a priori, okay, the things that line up with what you see epidemiologically are more right-sided tumors, younger onset patients, those type of things. But the things that don't line up is poor prognosis, where these have better prognosis, okay? So we looked at the other variables. So if you look at mucinous, these tumors lined with mucinous as expected, poorly differentiated, were higher than the microsatellite stable ones. More than half these tumors had this Crohn's-like reaction that I mentioned, and 81% were right-sided in this population-based cohort. But the MSI tended to segregate to white patients, not the Black patients. And that was statistically significant. We also looked at whether there's any difference between white MSI high tumors and Black MSI high tumors. And the bottom line is no. We saw a slight difference in age, but didn't reach statistical significance. The numbers get small because it's only 15% overall. It trended toward more Black females. And this is true for sporadic MSI. It tends to be more in females than males. But it trended toward it in our study. And there was no difference between the two MSI groups otherwise in this study. Then we looked at microsatellite stable. I said, okay, we got all these MSIs. Let's look at the stable ones. There's a difference there. And this is what's known in the literature too. So first of all, the Black patients tend to be younger and slightly more female, okay, and much more on the right side, as expected. So it's kind of matched what we thought we would see, except for the level of MSI. So this is Hassan Ashkatab. I mentioned him before. He put together a series of studies, two of which are from us, but there's not very many studies out there. And he came up with an odds ratio of African Americans having MSI tumors at 0.78, but it crossed the 1. And it's because of the number of studies. So it looks like it trends to be less in African Americans. And you just need more numbers, I guess. What about other levels of the immune profile? So this is a study this is the same patients from that study in which we looked at, okay, what's the level of tumor infiltrating lymphocytes? Because maybe the MSI is slightly different, but maybe it's the level of inflammation. Very interestingly, so if you look at MSI versus MSS overall, yes, the level of T cells in MSI, this is counting per high-powered field. You can have a computer do this, or you can have a a student count this if they have some time. But it was essentially 2 to 3 times higher if you had an MSI high tumor versus an MSS tumor. But if you look at African Americans versus Caucasian Americans for MSI, there was no difference for MSI or MSS. However, it was interesting, and you count all these tumors, and this is like 500 some tumors, there was no high level of inflammation. That's depicted by where this bracket is in the African American group, meaning you think they would have high levels of inflammation, but there were very few people, if any, that had these high levels of inflammation. So we don't know why that is, unless there's some other defect in the level of how the inflammation works. The other thing is, if you look at the microsatellite stable tumors, we looked at granzyme B. Granzyme B is a cytotoxic granule in white cells that can kill other cells. And if you look at whether you look at the intraepithelial lymphocytes or the intratumoral lymphocytes, the level of granzyme B positive cells was literally half of seen in Caucasians. Again, we don't have an explanation for this. It's just the number of cells. And this is the actual counts on these patients. So the other thing too, and this is from Luis Diaz, PD-1 blockade may improve the already beneficial survival of MSI high patients. One key question is, which we don't have an answer to, if African American patients have lower MSI high frequency, will PD-1 blockade improve outcome as a group or via individuals? I would suspect it would, but just if the percentage is slightly lower, there's going to be less people who can take advantage of this. So, but no one's done that kind of study yet. Now let me tell you about another aspect of mismatch repair, and that's called EMAS, or inflammation-associated microsatellite alterations. Let me see. Okay, I'll try to close up quick. I just looked at the time. I'm getting so much into this that I'm enjoying this. I mentioned that these mismatch repair proteins work as heterodimers, and these arrows show how they work. But if you have MSH3 defect, this perfectly overlaps with what we call EMAS, which stands for elevated microsatellite alterations at selected tetranucleotide sequences. So if you see EMAS, it's an MSH3 defect. That's the only way biochemically you can do it if you don't have mononucleotide or mononucleotide frame shifts. So you can predict which mismatch repair genes would be absent or present based on the biochemical assay you do. And so now MSI high was only done with mononucleotide and dinucleotide. We do all our assays all the way through tetranucleotide now, so you can quickly figure out what's going on. This was some early data we published just several years ago. We were noticing that when we stain cells for MSH3, and by the way, since MSH3 was not one of the major mismatch repair proteins, there were very few tools, very few antibodies and everything, so until they came online. But if you take out MSH2, you're not going to get MSH3, so that's a negative tumor. But interesting, in those who had this tetranucleotide instability, and I'm sure this is probably not projecting very well, there was some nuclear heterogeneity, as if the nucleus lost some expression, some cells lost it, some didn't. And it's like, what the heck's going on? And then the other thing too is this associated with IL-6. So if you stain a bunch of tumors and looked at the tetranucleotide instability, if they didn't have EMAS, you didn't find much IL-6, but if you had EMAS, you found associated with IL-6. So this led us to like, what is going on? By the way, no one could find germline until recently. There was no germline mutation in MSH3, and there was no mutation or epimutation in MSH3, even though we're finding this tetranucleotide instability. So what the heck was going on? Well, Stephanie Zeng-Rogenski in my lab figured it out. Turns out nothing was going on with the protein other than it moved location. So why did we get the heterogeneity? Because it went from the nucleus to the cytosol. Let me show you this. So this is IL-6, this is control, so no IL-6. The green is stained for MSH3, the API stains the nuclei, you overlay them, all the MSH3 is in the nucleus. You give IL-6, the green disperses, and overlap that with the nuclei, you see all the green is now outside the nucleus. You can see this biochemically here. This is taking an egg, taking the yolk, separate from the egg whites. So the egg yolk is the nucleus, and you can see with increasing doses of IL-6, it drops in that yolk or nucleus, and it picks up in the cytosol. This is not true for MSH6, MSH2, and MSH1. Only MSH3 is the one moving. And while it's out the nucleus, you take a genetic locus as a tetranucleotide sequence. While it's outside the nucleus, you start getting more mutations. This is just by inflammation, okay? So the way this works, without showing you a lot of data, MSH3 can move, and we've now mapped the nuclear export signals and the nuclear localization signals of MSH3 because they weren't previous mapped, and that's a paper under review. MSH3 can move out and back into the nucleus, unlike the other mismatch repair proteins. Amazing. I'm going to skip this next one. So the way we think of this is you can induce neoplasia. Who knows what's causing inflammation? Inflammation occurs. Now you get MSH3 shifting out of the nucleus. You can get double-strand breaks or other potential mutations. But the biological consequence is increased metastases. By the way, this is seen in half of all colon cancers. This is more common than MLH1 hypermethylation. But it's a somatic event. Some features, observed in 50%, they display EMAS. The tumors appear distributed evenly throughout the colon. They are non-hyperemitic. These are not MSI high tumors. They associate intimately with inflammatory cells. That explains the IL-6. And by the way, we did look at other cytokines. It was IL-6 who did this. They are sensitive to 5FU, probably because MSH6 is intact. They show shorter survival, unlike the classic MSI, and poor prognosis and more metastases. So they behave differently. And I'll show you, there's some data that's higher frequency amongst African Americans. This is data not from our group showing that, look at the survivability for MSI high. This is the EMAS curve. Just the opposite, okay? This is data from our group we published several years ago. Blacks, this was rectal cancers, by the way. So blacks had 49%, whites had 26% EMAS. 62% showed with VAT stage, and 60% associated with that classic inflammation. I'm gonna skip this. I'll just close out by saying, like, okay, so we got all this information, how are you gonna do this? Do we use anti-IL-6? Do we whatever? And so this is data that we haven't published yet, but we went back to our friend Bob Sandler in North Carolina, who had all this data, said, can you tell me who was taking some aspirin or NSAIDs? And he had all the data. And so we analyzed it. And it turns out that if you're we know that aspirin can reduce your risk for colon cancer and be a secondary prevention if once you had colon cancer. Turns out it gets rid of EMAS as well. And so if you were long-termers, your odds ratio on a multivariate was 0.26, based on our study. And it only worked in the proximal colon for a reason. So we don't know why that is yet, because the distal colon, we did not see that. So let me just quickly summarize and close out. So you can have MSI or MSS tumors. MSI gives you good survival. If you have an MSS tumor that gets the cytosol sequestration of MSH3, it seems to shift toward poor survival. The good survival seems to be slightly more common in whites. Right side of the colon, immunologically driven inflammation, PD-1 responsive. The EMAS seems slightly higher in blacks. Distribution in colon seems to be even. That's inflammation-initiated MSH3 dysfunction. That's sporadic and might be aspirin or NSAID responsive. I'm going to skip this. There are some disparities in colorectal cancer. African Americans with colorectal cancer, early age onset. We have these biomarkers I mentioned. There's reduced T cells expressing granzyme B, higher numbers of right-sided polyps that match the cancer. There's a lot of evidence of patient navigation, so getting ahead of the game works. And for the first time in 2017, the Multi-Society Task Force recommended an earlier onset for that. So I'll stop there. I'm sorry I went a few minutes over. And take questions.
Thanks so much, John. That was a big picture for colon cancer. Appreciate it. Questions for our guest? We'll give you a microphone if you don't mind.
Nice talk. I was wondering the right-sided the blacks have more of right-sided colon
Yes.
CRCs. Do they also show features of more hyperplastic polyps or serrated, which is more associated with the BRAF and SIM with the
Correct.
right-sided?
So the question is, could the difference be serrated polyps, which are higher risk? About 10% of those can go into colon cancer compared to a classic regular adenoma. The answer is there's not good data. So there was one data by Paul Schroy that looked at the level of serrated polyps. It was in that particular study, it was Annals of Internal Medicine, 0.3% in blacks, 0.2% in whites. So it wasn't much different at all. So they're not known to be all serrated adenomas. We just published, again, with Hassan Ashkartab, a group of serrated adenomas found in blacks. Now, this does not address the epidemiology. There seems to be some aggressiveness differences, but it doesn't address the epidemiology. So the answer is there's no evidence that these are more serrated in African Americans versus non-African Americans to date.
May I ask another question here? So the other thing is the EMAST. I didn't understand that. So that should also induce more microsatellite instability, right?
So no, it could, yes. So the definition of microsatellite instability is mononucleotide and dinucleotide instability. That's the definition from the NCI from that workshop 20 years ago. So EMAS is tetranucleotide instability. So when you take out MSH3, you will get some dinucleotide as well as tri and tetranucleotide. You will not get mononucleotide instability. So if you're only using classic MSI markers, which are mostly, a lot of people screen with mononucleotides, some will add some dinucleotides, you will miss tetranucleotide instability.
Interesting, yeah. Great, thanks a lot.
So we use a panel in my lab that gets all of them. So we can instantly tell you, is it MSI high, low, EMAS in one fell swoop. And we have it all multiplex. So we'll see if some company's trying to steal it from us.
It's cool.
From an epidemiologic perspective, are there any risk factors that are more associated with right side versus left side colon cancer?
There's
Other than race.
theories.
Yeah.
Theories. There is some evidence, in fact, from Hopkins here, Cynthia Shears with the microbiome work. So she showed last year.
Cynthia's here.
Oh!
Just let you know.
Duh! You can explain. I was going to mention some of your FAP work that goes throughout the colon, but some of the things that are probably more down the line, like Fusobacterium, seems to be on some of the right-sided tumors. I didn't show some of the preliminary data we have. Fusobacterium has been associated with right side of the colon. Okay, it seems to be in cancers, not polyps. And it has been associated by a pseudogenous group with MSI high colon cancers. We also have data that shows it's not just MSI high, it's also EMAS. Now, I can say, well, you just told me MSI is good, EMAS is bad. How could it be with both? And so we have a theory. We have the theory that there's a direct effect of the Fusobacterium, and we're doing some studies now that causes the IL-6 and the MSH3 shift. That's a direct effect. But we think the MSI associated is an indirect effect because Fusobacterium can generate reactive oxidation species, damage DNA, and then you can get more methylation, which can take out MLH1, and then you get an MSI high tumor. Now, that's a lot to theorize, but that's what we think. I'll just leave it at that.
So I'll make a comment. So that's fascinating. I'll add just two things. There's also these structures, biofilms on the right side of the colon that seem to be present in virtually 100% of tumors in that location for reasons that are unknown.
Yeah.
And something that's under review and so not out yet is that using the clinical practice research database from the UK, there's an association with prior antibiotic exposure and increased right colon cancer, but not left as well. So we may begin to see other factors that may link to the microbiome, as John mentioned.
Yeah.
Great
Thanks,
talk.
Thanks, Cynthia. Didn't see you there.
So I have to ask the question. So experimentally, we can drive a huge number of small intestinal tumors in any
Yes.
number of rodent models. I mean, that's
Absolutely.
It's really easy.
We've cured cancer in mice many times.
So why don't we see this in humans?
Well, there's, again, a lot of theories, and I think it has to do with both the physiology and probably the microbiome and the secondary products of the microbiome. So the small intestine, it has a huge surface area compared to the colon, but it's I wouldn't say constantly moving, but a near constantly moving organ. So things are flushed through. So the dwell time for any fecal contents in there is on the order of minutes to hours, whereas the dwell time in the colon is hours to days. If you do, we do like colonic motility studies where we give people markers and things over, or we give the capsule endoscopy. You'll get down to the cecum within hours, but that capsule could sit in your colon for the next 3 or 4 days, or longer, depending on your colonic inertia. So I think some of it is the rapidity of exposure and the time for exposure. I think some of it, the level of the microbiome is different in the colon than the small intestine. So that we have some in the small intestine, but it's much greater in the colon by a factor of 10 to 100-fold at least. And we see this when we have small bacterial overgrowth, when someone has an incompetent ileocecal valve or any other condition that may have removed their ileocecal valve, and they'll get bacterial overgrowth, and then it interferes with their digestion. So you have to have the level of the microbiome to a low enough level so it doesn't interfere with the nutrient assimilation process in the small intestine. There's also secondary products. We have things that are reclaimed in the ileum before it gets to the cecum. I mentioned earlier the intrahepatic circulation and how we reclaim the majority of our bile acids, and only a small percent leak into the colon. And of course, the ones that do, the bacteria can do lots of things with it. There's probably a lot of things. So it's probably exposure time, probably the local environment, and the products that are available at that time, and the dwell time that's in there. So these are all theories. No one's proven all this. In mice models, as you mentioned, a lot of the FAP models, it's all intestinal. There are some specific that have the right mutations that give you colonic tumors, but a lot of them are the small intestinal tumors, which we just don't see very much in.
If inflammation is so important, is there any benefit in just measuring fecal inflammatory markers, cytokines or other markers?
So probably not specific for colon cancer. So the things that come out in the feces can, most of the bacteria that most of the DNA that comes out in the feces are bacteria DNA. I mean, by factors of huge. So when we even when this, you heard about this Cologuard, I don't do anything with Cologuard. I'm not on the exact sciences or anything, that they have a whole process to separate out the bacterial DNA from the human DNA to get to what they're trying to assess. But inflammatory markers in the bowel might not be very specific to the large bowel at all. You can pick up KRAS mutations, and this is not even inflammation, from someone with pancreatitis. Pancreatitis, not pancreatic cancer, pancreatitis. So I don't think it's going to be good for just solely looking at the colon. And we have this balance and tolerance between the immune system and the microbiome in their colon that's always there. And then when you get the dysbiosis or something perturbs that immune system, that's when things go haywire. But I don't think it's going to be very specific if you did that specific for colon or colon cancer.
We'll draw out this session to a close, but let's thank Dr. Carethers one more time.
Thank you.
So for you who can stay for pizza, it's in the gallery, and
pizza.
Dr. Carethers will be out here to entertain some additional questions.